Dear children, I am here with another video of solving biology sample paper. So this time I have taken the sample paper 1. So let's get into the solutions of this paper. Now moving on to section B. Write the role of, actually this is origin of replication, okay, and restriction site in a cloning vector PBR 322. So if the term comes restriction, cloning vector, the chapter is biotechnology. Let us move on to the chapter biotechnology. Chapter biotechnology, principles and processes. So let's refer the figure given there, PBR 322. That is a cloning vector. You can see the figure here. So this figure is very, very important. You have to learn all the terms, all the labels here. So origin of replication. So this is the origin of replication ORI. And the restriction sites they have asked for. So first we will mark the origin of replication. So origin of replication, as the term says, it is the sequence from where the replication starts. Okay. So this is the sequence from where the replication starts. You can see here. This is a sequence from where the replication starts, and any piece of DNA when linked to this sequence can be made to replicate within the host cell. Anyway, whenever you learn this part, learn the whole thing. This sequence is also responsible for controlling the copy number of the linked DNA. So how many number of copies to be produced. So if one wants to recover many copies of the target DNA, it should be cloned in a vector where the origin supports high copy number. So according to that, we have to use, if the origin of replication, that is ORI, supports high copy number, more number of copies, we can use that. According to our requirement, we can select the plasmid with the required origin of replication. Now coming to the next part of the question, next question they have asked for the restriction site. Restriction site is nothing but the recognition site. So you can see the recognition sites here. Recognition site is the site where the restriction enzyme has to act. It is a site at which the restriction enzyme acts. So here we have the restriction recognition sites here, and here actually the restriction enzyme, they have mentioned the names of the restriction enzymes here, okay. E. coli R1, CLA1, HIN3, BMH1, SAL1 and PST1, PVU1. These are all restriction sites. These are all restriction enzymes which cut at these restriction sites. So restriction site is a site where the restriction enzymes, or recognition site is a site where it cut. It's a cloning site. It can be otherwise called as a cloning site. Okay, and this site is used to link the alien DNA. If you want to link an desired DNA here, we could use this. We could use any of these restriction sites. Clear? So in this figure, they have clearly mentioned here E. coli cloning vector PBR 322 showing restriction sites. These are all restriction sites. Okay, which is otherwise called as a recognition sites or cloning sites. The question, what is the role of? So we have mentioned the role of this both. The role of restriction site is to, what is that? It is to recognize the restriction enzymes, so that restriction enzyme cuts, and so that we can link the alien DNA. The vector needs to have, you should keep this in mind, a vector needs to have very few, preferably single recognition sites for commonly used restriction enzymes. And so if we are using a restriction enzyme, for example, if we are using EcoR1 should have one restriction site. So it can avoid confusions. Multiple cuttings can be avoided by using this. Clear? It is generally observed that children who had suffered from chicken pox in their childhood may not contract the same disease in their adulthood. Explain, giving reasons, the basis of such an immunity in an individual. Name the kind of immunity. So this kind of immunity we are acquiring during our lifetime. So definitely it will be an acquired immunity. And is it active or passive? Which chapter is it? Yes, human health and diseases. Let us get into that chapter. Human health and diseases. And they are talking about immunity, so definitely it is in the immunity part. It comes here, the acquired immunity comes here. Acquired immunity is pathogen specific. It is pathogen specific. It is characterized by memory. This means when our body encounters a pathogen for the first time, it produces a response called primary response, and then which is of lower intensity. Subsequently, encounter with the same pathogen elicits a highly intensified secondary or anamnestic response. Remember this word, okay? Anamnestic response, or which is a secondary response, that is once when the same pathogen attacks us, our memory cells will be active and they will be producing the specific antibodies. So that is called as a secondary or anamnestic response. So this is ascribed to the fact that our body appears to have the memory of the first encounter. So now let's read that question again. Here, the children who have suffered from chickenpox in their childhood may not contract the same disease in their adulthood. Explain, giving reasons, the basis of such an immunity in an individual. So this is the kind of immunity, acquired immunity. And also name the kind of immunity. It is acquired immunity. But one more thing you can mention here. If it is active acquired or passive acquired. So acquired immunity, we have two kinds, that is active acquired immunity and passive acquired immunity. If the kind of immunity which we acquire is naturally produced by the host body, that is, see, when a host is exposed to antigens which may be in the form of living or dead microbes and other proteins, antibodies are produced in the host body. So here in this case, once when the chickenpox virus attacks us, our body responded. That's a primary response. It is not that intense. So the body gets that disease. And second time, then the body will have the memory cells. So the second time when the same virus attacks us, we'll have the memory, and secondary response, that is anamnestic response, will happen, and that will protect us from the disease recurring in our body. So this kind of immunity is called as active immunity. So when they ask for naming it, it is active acquired immunity. Name the kind of immunity. It is active acquired immunity. Hope it's clear. What are interferons? Mention their role. Interferons, they are cytokine barriers. You remember cytokine barriers? Let's see the same chapter. You can see here. Cytokine barriers are actually special kinds of proteins. And one example of cytokine barriers are interferons. They are produced by the virus infected cells. Virus infected cells secrete such things, proteins called interferons, which protect non-infected cells from further infection. Got it? Now let's see the second part of that question. Mention their role. Yes. Their role is to protect non-infected cells from further viral infection. Hope you understood the answer. Next question. Study the given diagram. What do you think the diagram is? Yes, it is reproduction. Implantation. We'll see the chapter. The chapter human reproduction. The same figure is given in the textbook. It's here. You can see this both diagrams. Actually, they have asked this both diagrams, right? So study the given diagram. I think I've placed the question here. Study the given diagram. See the question once again. Study the given diagram. A is an embryonic stage that gets transformed into B. So what is A and what is B? Here, the answer comes here. The morula continues to divide and transforms into blastocyst. As it moves further into the uterus, the blastomeres in the blastocyst are arranged into an outer layer called trophoblast. Now this is actually morula and this is blastomere, okay? The first stage is morula and it forms the blastocyst. And in the blastocyst, we have two layers, that is arranged into an outer layer trophoblast. This outer layer is trophoblast. And an inner group of cells attached to the trophoblast called inner cell mass. So this one, inner cell mass, and outer layer is trophoblast. So what's the question? A transformed into B, which in turn gets implanted in the endometrium. So at this stage, at the stage of blastocyst, they get implanted. Identify A and B, that is A morula and blastocyst. C is inner cell mass and D is trophoblast. State the fate of C and D in the course of embryonic development in human beings. So what happens to the trophoblast and what happens to the inner cell mass, that's given here. The trophoblast layer then gets attached to the endometrium and the inner cell mass gets differentiated as the embryo. So trophoblast help the blastocyst getting attached to the or implanted in the endometrium, and the inner cell mass gets differentiated as the embryo. So this is their fate, okay? And that process, like blastocyst getting embedded in the endometrium of the uterus, is called implantation, and it leads to pregnancy. Hope it's clear. Coming to the next question, draw a pyramid of numbers considering a big banyan tree supporting a population of insects, small birds and their predators. So actually here they've asked to draw a pyramid of numbers. When we consider the pyramid of numbers, banyan tree would be small, like only one banyan tree, okay? Then supporting a population of insects, the population of insects would be more. The number of banyan tree will be less, so we'll draw one here, and just above that the next block will be representing this population of insects. So population of insects would be more compared to the banyan tree, only one banyan tree, because they've asked a number. They did not ask the biomass, they've asked a number. As they are asking for the number of like pyramid of numbers, so banyan tree is only one in number, so the base one banyan tree here, the block of banyan tree at the base, the number of, that is the population of insects, number of insects residing on the banyan tree would be more than that, right? So the number, the block showing the insect population will be bigger than this banyan tree. And if you just check the small birds eating on the insects, will be, the number will be lesser than the insects, but more than the banyan tree. Small birds. And number of big birds, that is the predators or eating over or feeding over the small birds, will be less than that, but more than the banyan tree. So the number of the pyramid to represent the number of these levels, that is banyan tree, population of insects, small birds and their predators, would be like this. Clear? Since it is number, if it is biomass, it will be looking different. Next question is, name the type of food chain responsible for the flow of large fraction of energy in an aquatic and a terrestrial ecosystem respectively. So aquatic and terrestrial ecosystem, and one difference between the two food chains. So let us check the textbook. So in chapter ecosystem, exactly the same statement is given in your textbook. Yeah, it's over here. In an aquatic ecosystem, grazing food chain is the major conduit for energy flow or major supply of energy flow. And as against this, in a terrestrial ecosystem, a much larger fraction of energy flows through the detritus food chain than through the grazing food chain. Okay. So in aquatic ecosystem, grazing food chain is the major conduit of energy flow, because the energy flow starts from the producers, like the phytoplanktons. So phytoplanktons are eaten by the zooplanktons, and that kind of grazing, that's what they mean by the grazing there in aquatic ecosystem. Whereas in terrestrial ecosystem, a huge amount of energy is released by this, energy is supplied by this grazing food chain. Much larger fraction of energy flows through this, not grazing food chain, detritus food chain, that is DFC. Okay, DFC, that begins with the dead organic matter. They give more large fraction of energy to the plants. So here in terrestrial ecosystem, detritus food chain, that is DFC, gives more energy, whereas in aquatic ecosystem, GFC gives more energy. Now they have asked for the difference between, detritus, that is DFC, and GFC. That's the next part. Mention one difference between the two food chain. So you know, detritus food chain begins with the dead organic matter, whereas grazing food chain begins with the producers. Okay, and it is made up of decomposers, which are heterotrophic organisms, mainly fungi and bacteria, and their energy requirement is satisfied by decaying dead organic matter or detritus. Whereas in grazing food chain, the The food chain starts with the producers, the phytoplanktons, then eaten like grazing means it can be in both terrestrial and aquatic. Here in the aquatic, the grazing food chain starts with the phytoplanktons, then zooplanktons, then fishes, and it goes on like that. Clear? Next question. Two children, one with blood group AB and the other group with blood group O, are born to parents where the father has blood group, it is actually A, and mother has blood group B. Work out a cross to show how is it possible. We know that blood group A can have both either homozygous or heterozygous condition, right? So here the children, one of the children has blood group O, so definitely it will be a heterozygous condition, isn't it? So you can represent as IA small I for father and mother IB and small I, and you have to use this as a parents and do the cross. So the gametes are small I, IB and small I. So what are the possible offspring? IA, IB, IAI, IBI or II. So this one can be of AB group, this one can be of A group, this one can be of B group, and this one can be of O group. So two children, one with blood group AB and the other one with blood group O is possible here. If both the parents are of like homozygous condition, then this will not be possible. That is, blood group O is not possible. Or if one of the parent is homozygous and the other one is heterozygous also, this group, O group, is not possible. So only if both the parents are heterozygous with one recessive each, the recessive O group will be appearing in the next generation. Is it clear? Now, coming to the next question. Carefully examine the structures A and B of pentose sugar given below. Which one of the two is more reactive? Give reason. So I have taken the figure. I'll just enlarge the figure so that it will be easy to. You can see here. This is deoxyribo, that is DNA, and this is ribonucleic acid. Okay, ribo sugar and deoxyribo sugar. And here, how the position starts? The position starts from here, position 1, it's carbon 1, carbon 2, carbon 3, carbon 4 and carbon 5. That's how it comes. The figure is clear. Carbon 1, 2, 3, 4 and 5. So here this is 1, 2, 3, 4 and 5. 1, 2, 3, 4 and 5. Clear? And now in this figure, it is clear that in deoxyribo sugar, the second position is having hydrogen, and in in ribo sugar, that is for RNA, the second position is possessing hydroxyl group. You understood? So this is A is ribose sugar and B is deoxyribo sugar. You understand? So that's how the orientation comes. Now let's get back to the question. So you understood this as ribose sugar and this is deoxyribo sugar. Now, so we have identified it. Which one of the following is more reactive? Give reason. Which one of the following is more reactive? Ribose sugar is more reactive. Let's see the reason. Chapter Molecular Basis of Inheritance. So we have taken molecular basis of inheritance. Let's see where the answer is. It says why RNA is more reactive than DNA. Do you remember that part? Why is RNA more reactive than DNA? They have explained it, yes, here. In RNA, 2 OH group is present at every nucleotide in RNA, and it is reactive and it makes RNA liable and easy degradable. And so it is known to be catalytic and hence reactive. So RNA is more reactive. And therefore DNA is less reactive. Because of the presence of H group there, it is less reactive when compared to the RNA, structurally more stable when compared to RNA. Therefore, among two nucleic acid, DNA is better genetic material. So the question can be asked in the other way too. Why RNA is, RNA is not considered, or DNA is considered to be the genetic material. Why have we come to the conclusion of why nature selected the DNA to be the genetic material? Got the answer? Yes. Next question. Explain the role of pituitary and ovarian hormones in the menstrual cycle of human females. Actually, you have to critically think and write this answer. It is not directly given in the textbook, but we'll just analyze and find it. Okay. So it is the menstrual cycle. So obviously, reproduction in human beings. Let's get to the chapter. Reproduction in human beings. Menstrual cycle with the graph given. So first you should know which are the ovarian hormones and which are the pituitary hormones. FSH and LH are the pituitary hormones, and estrogen and progesterone are the ovarian hormones. So that you have to understand first. FSH and LH are the pituitary hormones, and estrogen and progesterone are the ovarian hormones. And then you can write the role of FSH and LH. The secretion of gonadotrophins, that is LH and FSH, increases gradually during the follicular phase. This stimulates the follicular development as well as the secretion of estrogen by growing follicles. Both LH and FSH attain a peak level, and rapid secretion of LH leads to the maximum level, and it urges or it stimulates the release of ovum or ovulation. Okay, so FSH stimulates the follicular development and LH stimulates the release of ovum, that is ovulation. Now coming to the ovarian hormones, that is estrogen and progesterone. Now the role of estrogen. Estrogen supports ovulation, that is the rupture of Graafian follicle, and as well as meanwhile this endometrium, simultaneously the endometrium of uterus regenerates through proliferation, right? Before the secretion or before the emission or coming out of progesterone, estrogen will help in the proliferation of endometrium, and once the progesterone comes out, estrogen like progesterone will further supports the body or supports the uterus for pregnancy, prepares the uterus for pregnancy. Okay, and yeah, it's essential for the maintenance of endometrium, for the implantation and all the other events of pregnancy. This progesterone will be playing a major role. So these are the roles of ovarian as well as the pituitary hormones. Now coming to the next question. Differentiate between autogamy, geitonogamy and xenogamy. Can you guess which chapter it is? Yes, sexual reproduction in flowering plants. Let us get to the chapter. Actually, it is referring to the pollination part. Okay. Sexual reproduction in flowering plants. As I said, it comes under the pollination part. So let us take that pollination. Kinds of pollination. They have asked for autogamy, geitonogamy and xenogamy. Anyway, we'll learn all the three gametes in detail. So autogamy, in this pollination, actually it is the self-pollination type, that pollen grains will fall into the same flower. Okay, and if it is a same flower, even though if it is autogamy is possible in the same flower, see there are certain unique things they have given here. It can intricate things given here. It can be asked for some questions. So let us go through that too. In this type, pollination is achieved within the same flower. Okay, so one flower, the pollen grains will be falling into the stigma of the same flower. Transfer of pollen grains from anther to stigma of the same flower. In a normal flower which opens and exposes the anthers and stigma, complete autogamy is rather rare. Why? Because the flower is completely open, so there are chances for pollen grains from the other flowers to fall on the stigma. So we cannot completely claim that only autogamy will happen here, or the pollen grains from this plant can be taken away to the other flower. Autogamy in such flowers requires synchrony in pollen release and stigma receptivity. That is, only if both pollens and stigma matures at the same time, we could claim that only autogamy is possible here. And also, all anthers and stigma should be lying close to each other. Yeah, their position is also important, so that self-pollination can occur. Some plants such as viola, that is common pansy, oxalis and common lina, remember the examples, okay, produce two types of flowers because these examples are asked as objectives. Chasmogamous flowers, where both flower, that is similar to flowers of other species with exposed anthers and stigma, where they completely open. And cleistogamous flowers, what happens there? The flowers which do not open at all. So cleistogamous flowers, the flowers will not be open. In that case, we can say that 100% autogamy is only possible. So when anther and stigma lie close to each other, when anthers dehisce and flower buds, pollen grains come in contact. So only autogamy is possible. So thus, cleistogamous flowers are invariably autogamous because there is no chance of cross-pollination. And they produce ushered seed set even in the absence of the pollinators. You understand? So that's about cleistogamous and chasmogamous flowers. And autogamy, if they ask for the differentiation, autogamy is a kind of self-pollination. Now, gaichnogamy. Gaichnogamy, in this, what happens is, there is a chance like open flowers, like chasmogamous flowers, the flowers, the pollen grains from the anther to the stigma of another flower of the same plant, okay? It's of the same plant, another flower of the same plant. Although gaichnogamy is functionally cross-pollination, it is of the same flower. So genetically they will be similar because the pollen grains come from the same plant. And totally cross-pollination is xenogamous flowers. That is, xenogamy is the totally cross-pollinated flowers, transfer of pollen grains from anther to stigma of a different plant. This is the only type of pollination where pollination brings genetically different types of pollen grains to the stigma. So whenever you learn a question, as I said earlier, learn the head and tail, learn the whole part of that chapter, or sorry, whole part of that question, so that it will help you to solve the other questions too, other question papers too. Clear? Let's move on to the next question. Convergent evolution and divergent evolution are the two concepts explaining organic evolution. Explain each one with the help of an example. Let us jump to the chapter evolution. Chapter evolution. In this question, they have asked for both convergent and divergent evolution, explaining with examples. So let us get to that. First, we'll talk about adaptive radiation or divergent evolution. See, divergent evolution or adaptive radiation means evolution happens in an area with a common ancestry, and those organisms will radiate to different areas and they lead a different life. They have evolved into a totally different species, but basically they will be having a common ancestral history or a common character. See, these Australian marsupials, they all have a common feature that they are all marsupials, but they are totally different or variant species. So this kind of evolution, we call them as divergent evolution. They diverge, okay, from a common place or common ancestry, they diverge. And one more example for divergent evolution is this Darwin's finches. So these finches, these kind of birds, they are all finches, but they have developed different types of beaks according to their foraging habitat, according to the food they eat, okay. So their beaks has transformed or beaks have got modified, but basically they are all finches. And it is called as adaptive radiation because they adapt, they radiate to other geographical areas, and according to that area they have adapted. So we can just explain evolution in this perspective, and then we call it as a divergent evolution. And how about the convergent evolution? We describe evolution in a different perspective. What we say here, again we take the examples of mammals here. We have two kind of mammals in this, like placental mammals and Australian marsupials. We have placental mole and the same kind of mole where they have developed a marsupial nature. Similarly, anteater, marsupial nature, mouse, marsupial mouse, lemur, spotted cuscus, and flying squirrel, again marsupial thing. So this says that there are chances where different species from different area, they converge to an area and they have developed a common feature. So this is a different perspective where we could think of evolution. Okay, two different perspectives we think of evolution. It is convergent and divergent, and this talks about convergent evolution and the previous one talks about the divergent evolution. Hope it's clear. Now coming to the next question. Name the plant source of the drug commonly called smack. How does it affect the body of the abuser? Yeah. Which chapter? Yes. Which chapter? Yeah. Human health and diseases. Coming to the opioids. You can see that. Drugs and alcohol abuse. Yeah, it's here. Opioids are the drugs which bind to specific. So this is the common smack. Opioid, it is obtained from opium poppy. So the question they have asked is which plant, right? Name the plant source. Yeah, exactly. It is opium poppy. And how does it affect the body of the abuser? How does it affect? You have to write two sentences. You can write the first sentence here. Like opioids are the drugs which is obtained from this. Smack is a kind of opioid. And it is obtained from the plant opium poppy. What it does is it binds to specific opioid receptors present in our central nervous system and gastrointestinal tract. And you have one more sentence here. Heroin, it's also, that's otherwise called as smack. Heroin is otherwise called as smack. Commonly called smack. So what it does, it is used as a depressant and it slows down the body function. So this both sentences you have to write. Okay. Now, second part of that question is, what is humoral immunity? Again, the same chapter, humoral immunity. We have two types of immunity, like based on the action, we have two types of immunity, like humoral immunity and cell-mediated immunity. So, humoral immunity, you can see, I'll take that part. It's here, humoral immune response. Humoral immune response is nothing but the antibody mediated one. Okay, acquired immune response which is mediated by antibodies, that is humoral immune response. That's what they have asked here. So you have to mention how the antibody mediated response happens. Then second type is cell mediated, that also you keep in mind, cell mediated immunity, that is CMI. Then what will be this? HIR. Okay, cell mediated immunity, it is done by the T lymphocytes. Humoral immunity response is mediated by the B lymphocytes. Okay, B lymphocytes are the one which produces the antibodies. Okay, that's given here. You can see here, B lymphocytes are the ones which produce the antibodies. Okay. Then cell mediated immunity, you know, where does this immunity is shown? Once when the organ is transplanted, like organ transplantation happens, you know, at that case our body will start reacting. If our body starts reacting, then it is cell mediated response. Fine. So that's why we take some suppressants, immunosuppressants to stop that. Clear? Next question. Let's move to the next question. What is colostrum? That's an option for this. What is colostrum? Why are breastfed babies likely to be healthy? Why? What is colostrum? The same chapter, colostrum. It comes under passive immunity, that is the yellowish fluid secreted by mother during the initial day of lactation, that is called colostrum, and it has the antibodies IgA and it protects the infant. It protects the infant from like it gives all the it acts as an immune booster to the infants, and this mother's milk help in all ways. It's a complete food for the human infants for the first 6 months. Okay, then malaria, typhoid and pneumonia and amoebiasis. So they have given here 4 diseases: malaria, typhoid, pneumonia and amoebiasis. So some of the human infectious diseases. Which one of these are transmitted through mechanical carriers? Malaria, first you have to analyze like how are these diseases getting transmitted. Malaria through mosquito, typhoid waterborne disease, pneumonia airborne disease, amoeba caused by amoeba, amoebiasis caused by amoeba, and it is carried by houseflies. So which are the both diseases carried by the houseflies, like mechanical carriers? Malaria and amoebiasis. Malaria is carried by mosquito, the organism, or the pathogen is Plasmodium. Amoebiasis is caused by the pathogen is amoeba, carried by houseflies. Now write the palindromic nucleotide for the following DNA segment. Next question, write the palindromic nucleotide for the following DNA sequence. So palindrome sequence will be, so here they have asked for the sequence, the complementary sequence. So it is C, T, T, A, A, G. So look at the sequence, it is a palindrome, right? Palindrome is the one which reads forward and backward similarly. So CTTAAG, CTTAAG. So that's kind of sequence is called as palindromic sequence. Name the restriction endonuclease that recognize this sequence. So EcoR1. Let's see the chapter biotechnology, and there you can see the restriction endonuclease which cuts this. So let's see the chapter. So let's check the question. Name the restriction endonuclease which recognize this sequence. Yeah, here it comes. So this sequence GAATTC is recognized by EcoR1, E. coli R1. So this palindromic nucleotide sequence is recognized by EcoR1. Now second question, how are sticky ends produced? Mention their role. Yeah, it also comes in the same part. Let's just check the textbook. You can see, so this is a sticky end. And how are these sticky ends formed? Restriction enzymes cut the strand of DNA a little away from the center of the palindrome sites, little away, that is from this part here, G next to G, and here just before G, but between the same two bases on the opposite strand. So here just after G and here just before G. This leaves a single stranded portion at the end. So you can see here it leaves a single stranded portion here G and at this base G. There are overhanging stretches called sticky ends. So this is how the sticky ends are formed on each strand. These are named so because they form hydrogen bonds with their complementary cut counterparts. So they stick each other by forming the complementary hydrogen strands. This stickiness of end facilitates the action of the DNA enzyme, like DNA ligase enzyme. So it helps the DNA ligase enzymes to do their role easily. Now coming to the next question. A particular species of wild cat is endangered. In order to save them from extinction, which is desirable, in situ or ex situ? Justify your answer and explain the difference between these two approaches. So what is in situ approach and what is ex situ approach? Do you remember? Let's see. Chapter Biodiversity and Conservation. So towards the end of the chapter, they talk about the conservation part, how to conserve the biodiversity. There, they talk about in situ conservation by constructing or by considering or creating biosphere reserves or announcing a certain area as a biosphere reserve or national parks or sanctuaries. So in all these cases, they conserve the animal or they protect the animal in their natural habitat. But in certain cases, if the natural habitat is not suitable for them to live, if special care has to be given, then we place in special settings where they can be protected and given special care. Okay, so in this approach, threatened animals and plants are taken out from their natural habitat and placed in special setting. So let's check the question again. They have asked for protecting a particular species of wild cat. It is endangered. And to save them from extinction. So it means they are threatened and a special care must be given. So we have to go for ex situ method, ex situ mode of conservation, and that's how we have to justify, where they can be taken out from their natural habitat, protected and given a special care. Okay, so it is more advanced, but still. Now what's the second part? The difference between these two approaches. So you have to write the difference between in situ approach and ex situ approach, how the in situ approach is done, like here it comes here. To conserve their biological wealth, maximum protection in biodiversity hotspot, that is in their natural habitat, we are protecting them. They are collectively protected in their natural habitat, where ex situ, they are taken out and given special care and special attention in a special setting. Got it? Now, next question. Coming to the case-based questions. Look at the diagram and answer the following questions. So, what do you see here? You can see a monohybrid cross. in a monohybrid cross, they have taken the heterozygous parents. So what type of genotype will be formed? So that is capital T, and small t, capital T, and capital T, small t. Okay. So you have to write the genotype and the phenotype of it here. 3 tall and 1 dwarf, and the phenotypic and genotypic ratio 3:1, 1:2:1. Okay, this is F2 generation of monohybrid cross. Write the conclusions Mendel arrived at on dominance of traits on the basis of monohybrid cross that he carried out in pea plants. I'll show you where the answer comes in the textbook. Please take Principles of Inheritance. Principles of Inheritance chapter. Please take that chapter. And after the cross, they have explained about the dominance, law of dominance. See here, they have explained here this part. You have to explain this part. Okay, characters are controlled. So what conclusion Mendel arrived? That's the question, right? We'll see the question again. What conclusion Mendel arrived at on dominance of traits on the basis of monohybrid cross that he carried out in pea plants? Characters are controlled by discrete units called factors. They occur in pairs. In a dissimilar pair of factors, one member of a pair dominates the other one. Villagers in a place near Chambur started planning to make power supply for agriculture purposes from the cow dung. They have started a biogas plant for the purpose. Study the flow chart for biogas production given below and answer the questions. Okay, so you wonder, actually this kind of chart is not given in our textbook, but they have mentioned about this biogas plant. So we have to use your critical thinking. There they have said that using all these base molecules, and this has to be converted into soluble compounds by facultative anaerobic bacteria. Then they will be subjected to fermentation. Actually, B will be fermentation with the help of fermentative microbes. And A, after this, methanogenic microbes will be used. And then biogas will be formed. Then you have to write the advantages of biogas. Okay. Next question. Describe the events of oogenesis with the help of schematic representation. It's a direct answer. Oogenesis. Let's check the textbook. Chapter Human Reproduction. You can see the diagrammatic representation of oogenesis. Both oogenesis and spermatogenesis are important. You must learn that both. You can see this, oogenesis and spermatogenesis. Now what's the next question? What's the second part of that question? Write two differences between oogenesis. You can write any two differences between oogenesis and spermatogenesis. So what are the two differences between oogenesis and spermatogenesis? First difference, you can write what the process is. That is, as a result of oogenesis, what happens? What is oogenesis? The formation of eggs. The formation of eggs. So what is the difference? What's the first point you can write here? You can write the definition. That the production of eggs or the formation of eggs from oogonia is known as oogenesis, whereas the formation of sperms from spermatogonia is spermatogenesis. Then it occurs in testes, it occurs in ovary. That process you can write. And then actually the spermatogenesis is a continuous process, right? Whereas the oogenesis is a discontinuous process. It is not continued at a stretch. Later on, it will be, the process will be stopped for a while, and later on, during the process of ovulation, it has been continued. So it is a continuous and discontinuous process. Oogenesis will be happening, the one part of oogenesis will be happening towards the later stages of life. So you can write that. And here, how many active or viable sperms are produced here, there how many are produced. So that kind of differences you can write. Fine. Now next question. When a seed of an orange is squeezed, many embryos instead of one are observed. Explain how it is possible. About polyembryony. Actually they are talking about polyembryony. Yes, here they are talking about polyembryony. You can see that you have to write what is polyembryony. We will read the question once again. When a seed of orange is squeezed, many embryos instead of one are observed. Explain how it is possible. So it is because of polyembryony. So you have to write what polyembryony is. In citrus is actually orange. and mango, varieties of some nucellar cells surrounding the embryo sac. So nucellar cells surrounding the embryo sac start dividing and they protrude into the embryo sac, developing into embryo. Here the nucellar cells are forming the embryo. In such species, one ovule contain many embryos. So this condition is called as polyembryony. And now they've asked you the next part. Are these embryos genetically similar or different? They are genetically similar. Obviously, they are genetically similar because they are all formed from the same nucellar cell. So, they are genetically similar. So, you have to mention that. Describe the process of synthesis of fully functional mRNA in eukaryotic cell. So, you have to write the formation of mRNA in a eukaryotic cell, and also you have to write the formation of mRNA in prokaryotic cell. Okay, so you have to mention the eukaryotic cell, the formation of mRNA, all the steps, all the formation, all the processes in the formation, and then you have to differentiate. Differentiate means you have to write how it is different from prokaryote. So better if you forget to write the differentiation, you can explain the formation of mRNA in prokaryotes. You can simply explain it. And the second, the option part of this is the given flowchart highlighting the steps in DNA fingerprinting. So this process is exactly the same. So I have taken this from the textbook. So this is the same from the textbook. And see, isolation of DNA from blood cells, cutting DNA by A. So A is restriction endonuclease. Separation of DNA fragments by electrophoresis using B. So it is agarose gel. Then you have to electrophoresis is by agarose gel, right? Next one, transfer of, or blotting of fragments to C gel. C gel, what we use here, nitrocellulose or nylon. Then DNA splits into single strand, and introduction of labeled D probe, VNTR probe we use there. E of single strands with D, detection of hybridized DNA fragments. So hybridized strands we get. E of single strands with D, hybridized DNA fragments with D, and detection of this hybridized fragments by F, it is by autoradiography. And then you have to write the applications of DNA fingerprinting technique. So coming to the next question, explain how to find whether an E. coli bacterium has transformed or not when recombinant DNA bearing ampicillin resistant gene is transferred into it. Shall we go to the chapter? Chapter biotechnology. How do we identify, or how do we realize that the DNA formed is recombined or not? So that is by the presence of that antibiotic resistance. So this part, okay, this part. How do we find that transformants has antibiotic resistance, like antibiotic, yeah, resistance. How do we recognize that the E. coli is transformed or not? Okay, so this is how we have to write, using antibiotic resistance. You can start from this example. You can ligate a foreign DNA at the BAMH1 site of tetracycline resistance gene in vector PBR322. Then recombinant plasmids will lose tetracycline resistance due to the insertion of foreign DNA, but can be still selected out from the non-recombinants by plating the transformants on a tetracycline containing medium. Okay, so this is the part. Fine. We can start from this. The ligation of alien DNA is carried out at a restriction site present in one of the two antibiotic resistance genes. So what they mean is, if they place, like if they place, or the if the foreign DNA is ligated or joined at the BAMH1 site of tetracycline resistance. So here the DNA has got ligated. So the transformant will not have tetracycline resistance. So now the recombinant plasmids has lost the tetracycline resistance because we have inserted it, but can be selected from. Now we have to know which are the transformed one and which are the non-recombinant ones by plating the transformants on a tetracycline medium. So when we place it on a tetracycline medium, so the recombinants will not have that resistance, right? So the transformants growing on ampicillin containing medium are then transformed on a medium containing tetracycline. The recombinants will grow in ampicillin but will not grow in tetracycline. And the non-recombinants will grow on the medium containing both the antibiotics. So in this case, one antibiotic resistance gene helps in selecting the transformants, whereas the other antibiotic resistant gene got inactivated. Okay. So this paragraph you have to write. And the next part of that question is, what does the ampicillin resistant gene act as in the above case? So that's what they have given here. The last sentence of that statement, that is, where in this case one antibiotic resistant gene help in seeing the transformants. It is antibiotic. The other one is inactivated due to insertion, that is tetracycline. Insertion of alien DNA, it helps in this is insertional inactivation. We call it as insertional inactivation. We have inserted the alien DNA or the required desired DNA, and that desired DNA has inactivated the tetracycline. So it is insertional inactivation. So here, this is the answer to this first statement. In this case, one antibiotic resistance, that is ampicillin. If it is having only ampicillin resistance, it means that it is a transformant. Okay, it has got transformed. Now here, this one, there are two different farmlands where one BT cotton crop was cultivated and the other where non-BT cotton were cultivated. Now farmers responsible for this experimental cultivation were free to use the farming practices of their choice. So during the cultivation period, the data was collected with respect to the amount of pesticide used, water required and harvesting time, and the crop productivity. Based on the data collected, a bar graph was plotted. Now looking at the bar graph, you know this light shaded one stands for BT with BT cotton, BT, and the other one without BT. Okay, so if we just check, the crop productivity is high in BT, pesticide usage is less in BT, but the water requirement is high in BT. So that's what we could interpret from this bar graph. Now write your interpretation with reason. Yeah, you can write your interpretation that crop productivity is high, usage of pesticide is less, water usage is more and all, based on the three parameters. So you can write your interpretation. Which one of the crop would you like to cultivate in your farm and why? Obviously we go for BT because the crop productivity is high and the usage of pesticide is less. Which one out of these two crops would a farmer from a West Bengal like to cultivate? So West Bengal they have emphasized because the water requirement here is high. West Bengal, there is no water scarcity. So in that case, they can go for BT cotton. But if the areas where there is a water scarcity, then they should think if they could use BT cotton or not, because relatively the water they use, irrigation should be high in the case of BT. So by that we have come to the end of this question paper. Hope it's clear. And by discussing this paper, you would have come to a conclusion, or you would have realized that how important is to read the textbook and to learn the textbook. Clear? All the best, children. Do well. We'll be meeting soon with another solution video. If you have any doubts, please comment. Thank you.